afternoon, everyone, and welcome to today's Public Health Ontario Microbiology Rounds presentation on whole genome sequencing, methods, utility, and implementation challenges for infectious disease surveillance. My name is Dr. Vanessa Tran, and I am a clinical microbiologist at Public Health Ontario, and I have the absolute pleasure of moderating today's session. Before we begin, I just want to mention a few housekeeping items. Firstly, the chat pod has been disabled to limit any distractions during the presentation. So please use the Q&A pod if you have any questions during the session. A discussion and question period will follow the presentation. At any point during the session, you experience any technical issues, please email capacitybuilding@oahpp.ca. I would like to state that as moderator of the session, I have some conflicts of interest to disclose. I am on the board of the Canadian College of Microbiologists and Canadian Association for Clinical Microbiology and Infectious Diseases. In addition, I am also a co-investigator or collaborator on grants that are funded by CIHR, CERN, PHAC, and Gilead. It is now my absolute pleasure to introduce the speaker for today's presentation, Dr. Aaron Campagato. Dr. Campagato is a medical microbiologist, division head of microbiology at SickKids, and assistant professor at the University of Toronto. His interest lies in the development of molecular-based clinical laboratory tests and the expanded role of next-generation sequencing within the microbiology lab to assist in clinical care. Now I'd like to pass it on to Aaron for a great presentation.
Thank you, Vanessa, and thank you for having me here. And hopefully, this will be a great discussion and great topic today to present. I'm very excited to go through this with you all. So in terms of the learning objectives, hopefully by the end of this talk, we'll be able to go through some of the challenges and methods and utility of whole genome sequencing and its use in infectious disease surveillance. And through this talk, we'll develop a approach and at least one approach to analyze and interpret whole genome sequencing data for, again, for different pathogens and appreciate the challenges in implementation of this sort of testing and its use in clinical and surveillance methods. So in general, when we consider whole genome sequencing in any public health surveillance or a hospital infection control program, there are multiple different ways that we can implement such a program. The program may be reactive, proactive, or reflexive. And what that really means is that we can be reactive. So we can really use this type of technology and this data to respond to a specific need. So for example, in a hospital or in a environment of your choice, you have a specific organism that you want to understand if cases are potentially linked at the genomic level, we could use that. But that is, again, a reactive response to a specific incident. We may use whole genome sequencing more proactively, and that is targeting certain populations, potentially vulnerable populations or certain organisms, to sequence all of these organisms in order to better understand the pathogen, the transmission potential, the virulence even, and understand the organism again at this genomics level. But in the proactive approach, we are doing this upfront without knowing potentially some of the epidemiological links initially. The last way can really just be reflexive, and that's almost an extension of the proactive approach. So again, we might not have as much clinical epidemiological information, but we are targeting specific pathogens to better understand the organism, their virulence, their distribution, and transmission potential. So it depends on what type of pathogen we are focused on, the amount of the pathogen we see, say, in the environment, and potentially even the pathogenicity of that organism. And we may choose to implement programs at any of these three different levels. Each of them also, I think, is important that they could be implemented at a public health and a broad level, or it can be implemented more locally at a hospital level, for example, within infection prevention and control. Overall, and I think this is something that I know I won't focus enough on, but it's very important to any of these programs, is the timely access to data and sharing of data. So with all of the data that is generated and all the information that can come from whole genome sequencing for a pathogen, it's important that we can share this data among public health units, among hospitals, among provinces, and even countries. So timely data sharing and timely access and testing is very key to the use of this type of technology. And really, what is the ultimate goal of the data that is reported from whole genome sequencing? And really what we're trying to do is determine if transmission has occurred between cases. We might want to see if there are certain types of strains of a pathogen occurring in a province or a region within a province or within a hospital, a unit within a hospital, for example. And this information is very powerful. We have used other types of methods within the lab for surveillance and within infection prevention control investigations in the past, but the information that we get from whole genome sequencing is much more powerful. And we'll talk about in the next few slides some of the important considerations of that extra detail that we get from whole genome sequencing. It's also important that whenever we use this type of method or any type of method within the lab for surveillance purposes, that we put this into context of the epidemiological situation. So laboratory data and laboratory methods and information is not or cannot really be used in a silo. It's important to put it into context with the overall clinical epidemiological picture and all of the other important investigations that supplement and support this type of analysis and work. So over the years, over the past many decades, we have been using other types of methods, not whole genome sequencing, to help and apply to pathogen surveillance. And we have used those for outbreak investigations, for organism identification, and general surveillance within a population. Those methods have been either phenotypically based, all the way up to the now whole genome sequencing. And just I've listed a few of those different types of methods, and I think many of you will be hopefully familiar with those methods. They are still used today. We still implement, we still understand how to interpret these methods for various pathogens well. And one of the key differentiations between each of these methods that we'll talk about in the next few slides is the differences between resolution of an organism and relatedness between organisms, and what type of information we can glean from the different types of techniques, and how whole genome sequencing in particular as a method is useful in some of these instances. So again, comparing the historical methods of pathogen typing and surveillance methods, we have used just susceptibility typing methods in the past. Very quick, very efficient method to use primarily for bacteria. We have moved that forward to pulse field gel electrophoresis, oftentimes for surveillance, for outbreak investigations, foodborne pathogens, AROs within hospitals. We have relied on pulse field gel electrophoresis for a number of years. So I think it's always important to put WGS into that context, because this used to be in some ways the gold standard. And then finally, whole genome sequencing. So you can see a list of various characteristics of each of these methods on the left-hand side, and two I really wanted to point out. One is the discriminatory power. And so the discriminatory power from whole genome sequencing is really what gives us the advantage and the change that whole genome sequencing allows. We can get a lot more information, and that will apply to some of the resolution that we are able to pull out that we'll describe. And the next piece is really the complexity of data and ease of use. So while pulse field and even all of these other different types of methods were sometimes relatively easy or less labor intensive, whole genome sequencing is more challenging to implement. And I think, you know, as the years go by, it will become likely more like an easier process, and more information will be able to be gleaned from that data, but it's still being developed. And whole genome sequencing is where it is today because of developments in the changes in the technology. So there has been a number of changes, and I don't want to spend a lot of time on those technologies in particular, but it's really been the evolution of our next-gen sequencing technologies that have allowed us to get to this point that have a rapid turnaround time, a robust process to have the entire genome of a bacterial, viral, or other fungal pathogen so that we can use this data in surveillance programs. And on the left-hand side here, you can see that over time, and I think we're all aware that the cost of this technology has decreased significantly. And so now we're at a point where we can implement in real time these sorts of technologies to better understand diseases. And finally, just again, in terms of that technology, I won't go into the specifics, but for those who are interested, there are many different types of technologies out there, a few different main differences in terms of long or short read sequencing. We won't go through that at all, but I think it's important to understand that regardless of the process that's being used, the output and the interpretation can be similar. It's important to also understand that at this point, unfortunately, there is some complexity in terms of the data analysis. And so that's where I want to focus our methods on and the interpretation piece. So in terms of whole genome sequencing, the major difference compared to other surveillance methods is that ability to have a better resolution of a pathogen. This is a nice example that the CDC presents for Salmonella cases as a foodborne pathogen within the US over a number of years. When we look at the left-hand side of the screen, we can see Salmonella cases over a 1-year period. They're all unlinked. We don't know. We could put them into groups based on clinical epidemiological information, but if we just focus on pathogen type and methods, there needs to be a way to try to link these different pathogens and these different organisms. So what we have used historically is pulse field gel electrophoresis. And you can see once we use this type of method in the middle of the screen, we start to get clusters. We start to get these groups of pathogens that look identical. And so we can see that this top group has a lot of these blue pathogens. The middle has some red and the bottom mainly yellow. And we, from a laboratory perspective, without considering the clinical data and the epidemiological data that accompanies each of these cases, we would assume that these cases are potentially linked. But once we move to whole genome sequencing, the resolution of the information, the detail that we can glean from the laboratory data is much more comprehensive. So now you can see that within this right-hand slide, many of the cases are no longer linked. And so we've ruled out that these cases are identical. We've ruled out that they are linked. We can focus our efforts on cases that do look the same. So we can see this very tight red cluster in the middle here and this other yellow cluster. So we can start to focus on cases that are linked. We can use our resources better. And we can also, we've ruled out transmission. We've ruled out certain cases. So that's what's the detail of information that whole genome sequencing provides. And it's important that we've, in all of these cases, essentially ruled out transmission patterns. We've ruled out cases. We haven't necessarily ruled in and shown that these cases are linked from a surveillance perspective. And so I wanted to give just one example here of a comparison and how those different methods have changed, a real life example. So this is some of the utility of this type of technique. So when we look at this case here, we have different methods of MRSA typing, so methicillin-resistant Staphylococcus aureus typing. And on the right-hand side of the screen, it has the least discriminatory, least resolution, called SPA typing. I think that the actual method is irrelevant. It's just the data that we get from the method. If you look at the middle panel, we have something called MLVA typing. So again, another molecular-based typing method. And on the left-hand side, we have whole genome sequencing. When we compare these methods, so SPA typing on the right-hand side, we can see there were 12 different groups, 12 different clusters of cases. This was over the course of an entire state in the US. In the middle, we have 17 clusters. So now when we focus on this bottom of the group, we can see those 12 cases now became 2 different distinct clusters. We've now ruled out that all of these patients, all of these individuals were related. And we can now focus efforts again on 2 distinct events. Moving to even more detailed information, we see whole genome sequencing. And what that has done was allowed to take these 2 different clusters and rule out cases. So we can see case 20, case 42, and this reference case to be very much different than others. So we've now, again, are able to get better resolution, understand transmission dynamics, understand the pathogen in a more detailed way within the lab to focus our non-laboratory investigations as well. So that was the one big advantage that whole genome sequencing provides from a surveillance perspective in terms of laboratory testing. The other main key defining term is relatedness. And this is something that I wanted to spend a little bit of time on because it's a very challenging concept that changes depending on the organism, essentially. And it's something that we're still defining. So in the past, again, moving to that gold standard pulse field gel electrophoresis, at least for bacterial comparisons and surveillance, we had defined criteria. We knew how to determine if organisms were related. We used something called the 10-over criteria. It was now a 20-plus-year-old document, but still used. When we take that concept to determine how different or how related are organisms based on their whole genome information, we're still in the early days of making those determinations. And it's so important in able to understand how we define relatedness and how we define transmission and surveillance programs. So one key concept is how quickly a pathogen will change, how many times we have a base pair, how many times we have some genetic change occurring in an organism. And that change is called a mutation rate or molecular clock. And every organism can be different. Bacteria, viruses, fungi, parasites, they all can be different. And it can also be different within those groups. So one virus may change quickly, another virus may change much more slowly. So instead of using one single set of criteria, we have to look at an organism level type of process. And that has to be defined in order to really use this sort of whole genome sequencing to define and look at these different pathogens. We also have to consider some of the lab challenges in terms of testing that may lead to false interpretations. So here's just one example. You can see here's one common ancestor. This is a genetic sequence. And we'll say in this organism, every 2 weeks, we get a new change in the genome. And so after 2 weeks, you can see we now have 2 different ancestors or 2 different organisms. Again, 2 more weeks go by, we have, again, 2 more changes. So after 4 weeks, we can see these at one point were related. Now they look different. They look different genetically. And how many changes do we need to compare organisms is the key. And it's not, you'll see that it's not, unfortunately, known for everything. So when we look at one example, this is norovirus, and it was trying to look at the number of base pair differences or similarities between different organisms to determine what is part of the same group or same cluster. And so when we try to do this in the lab, the first thing we would want to do is look at different genotypes. So norovirus has different genotypes. And we can see here from the left-hand side, we had a specific genotype, and over on the right, a different genotype, and the number of base pairs between those different genotypes that are different. What's nice, these light bars cluster together, the dark bars cluster together, meaning that the light group is related, the darker shaded group is also related. But within that genotype, how do we define relatedness? How do we define how closely similar are these viruses? So what we need to really do is we need to combine whole genome sequencing with the clinical epidemiological data. So we want to try to look at patients and individuals at a case level within an outbreak, and even at the same patient over time, to see how quickly and how many base pair differences, how many genetic changes occur. And so we can try to use that knowledge to start to define relatedness from a whole genome sequencing perspective. And it's still, like I said, it's still a very challenging process and something that is evolving. So now, once we have all this data, we know we want to look at the resolution of an organism or a pathogen. We know we can also try to define relatedness, but we want to use this information for surveillance. How will we do this? How will we communicate this information from a whole genome sequencing perspective to end users? So we may want to look at a whole genome sequence MLST or core genome sequence MLST. MLST, multi-locus sequence typing, is a method of molecular typing that has been used before whole genome sequencing. And in that case, we would look at 5 to 10 different genes within a genome. What whole genome sequencing has allowed is for us to look at many dozens, hundreds of different, thousands of different genes across the genome over time. And we look at, again, differences within that genetic code to say, are these two organisms potentially related? The challenge here is, again, that ongoing clinical validation and how to define what relatedness is. Another, and I think more commonly used method, is looking at whole genome sequencing using single nucleotide polymorphisms. So that essentially is trying to look at the genome from an individual level and look at each base pair within that genome. So in this example here, we can see this is Salmonella. These are different base pairs, different locations within the genome for Salmonella. And at any one spot, you can see the base pair, A, T, C, or G, may be different. So this one that's circled, all of these bottom organisms have a C in this position, while the top two have a T. So that tells us that there is a difference in the genetic code between these organisms. Is it significantly different? That's the next question that we need to answer. But at least we have looked and we can communicate to end users to say, genetically, we have a difference. Genetically, there's a different base pair at a certain position between these two organisms, and then use that as a way to communicate relatedness. And how do we overall look at that in terms of, say, a cluster of cases, an outbreak, or relatedness? We may want to use it in this very hopefully simple example. We have 4 different strains. We have these different base pairs represented by a different color. When we look at A and B, the colors are all the same. The genomic sequence is the same. So what that can tell us, based on the relatedness, is that these are possibly related. We can't say that they are genetic. We can say they're genetically related. We can't say that they're clinically, epidemiologically related, but we can say that they are genetically the same organism. When we look at A and C, again, we have a couple colors that are different. And so now we don't know if they're related, but we are more confident that they are possibly within the same group. And then finally, when we look at this organism D, so organism D has differences. We have one difference here. We can see that this one is, these 3 are blue. This is not. This has a green base. This has a purple. These do not. So there's lots of differences in terms of the genetic code for this organism D. So between D and A, B, and C, we can say genetically they are different. We can use that data to help us infer transmission. We can say transmission did not occur between these cases in an outbreak or in a cluster of cases. We can say that this pathogen is different. And that's really the only thing that we can say from this tree or from this group of genetic cases. That is still completely dependent on how many differences in that genetic code there are to make an organism different from others. So that's how we try to take that data, the genetic data, and look at relatedness. So I'm going to use the case of adenovirus over the course of these different methods. Adenovirus is an important virus that we see in the pediatric population within bone marrow transplant patients, and I'm going to focus on some data that was presented by the Greater Ormond Street Hospital in the UK on adenoviruses within their hospital context. So the first method would be to use genetic data. So here, if we used from a laboratory perspective, this sort of reported method, we are providing consensus genetic sequences for all isolates. So we are providing this genetic code for each of the organisms. And what's really an advantage here is that there's lots of data. It's raw data. We might not be able to define what's related. We might not be able to understand how to make those distinctions yet. So we are providing that data. If it's important to go back and incorporate clinical epidemiological information, all that data is there. The challenges that I really see is that it's really hard to parse out this data and interpret from a visual perspective. So I think many data analysis and informaticians and others may want this data, and it's a very powerful tool that should be available. For the general end user, I do see that it's personally something that can be confusing and challenging to interpret. And I think that was the least common response from the poll. The most common preferred method was a phylogenetic tree. So phylogenetic trees are very visually appealing. And so what a phylogenetic tree would allow us to do is visualize the relationship between organisms, pathogens, for example, and it is very much easier to look at these different organisms in a larger data set. The challenge that I can see here is that there are a number of different biases that can be incorporated, and then also the learning curve on how to read and interpret phylogenetic trees. For example, it is sometimes easy to confuse how to read the relatedness between these different organisms at the tips of this phylogenetic tree. So one important concept is that along these nodes, trees can pivot. So D and C are just as related to D and E because we can pivot that node between those different organisms. So it's understanding what is equivalent in terms of a phylogenetic tree and how to read that phylogenetic tree. So if we, as a laboratory, were to provide a tree, for example, I think it's important until they become more commonplace and used more frequently, that we do spend time sitting down and working through what the interpretation is and how to best utilize this sort of data. And so from a cluster of those adenovirus cases, you can see here's a phylogenetic tree. We would easily, I think, be able to visualize that we have 4 different clusters here within this one group of cases, because the phylogenetic tree shows that they are all along the same line, and it's a visually appealing method to see. And in that, there's a number of cases. There's dozens of cases, and it's still something that is able to be, that information is able to be transmitted to the end user. And then finally, the last method would really be to look at the a line list. So I think this is a very useful method. Many clinicians and end users, I feel, prefer this sort of method. It's easy to convey relationships. So we can, for example, have a typical line list in an outbreak. We can assign a genomic cluster, and based on that assigned letter or number, we know that there's a relatedness between those organisms. The challenge here is that it's up to the laboratory to define what is related. We can't necessarily go back and say, with more data and more understanding of the genomic differences to reanalyze, it goes back to the lab. So that end user has less of that raw data, less of the details, but much more, I think, much easier to just understand at a glance what is related and what's not. And so here's an example using that again, adenovirus case. You can see there were different clusters. So cluster 1, cluster 2, cluster 3. And so we had the genetic data, assigned number, the clinical epidemiological data, assigned a number, and how they determined that genetic relatedness based on the number of genetic bases that are different between pathogens. So again, easy to visualize, lots of information, but maybe more loss of some of the details and some of the power that whole genome sequencing can provide. And so in the last few minutes, I just wanted to go over some of the ongoing challenges to the use of whole genome sequencing that I do see. So one, wanted to first start on some of the wet lab and some of the laboratory processes that where some of the challenges exist. So first, if we have a pathogen that is in a mixed population, so we want to try to identify a virus within a patient, and there are 2 of that virus that are similar, we might not know which is at the higher amount. So you may not know, be able to have the detail of what is at and separate 2 populations within a certain patient or person. The other challenge is even when we use this for bacterial culture, a culture may be selecting for strains that grow better. We also may not sample multiple strains within a person. So even when we look for culture whole genome sequencing from a culture, it still may not represent the entire genomic population within an individual. And that may give us a false sense that this person is not related to others within an outbreak or within a cluster of cases. The other challenge that really is more of a laboratory issue in a number of ways is that standardization. So there's standardization throughout the process. There's standardization from the testing. There's also standardization in the informatics pipelines and the analysis of the data. So how much data is required? What is the quality of the data? And then we've already talked about at length the relatedness issue and how many mutations that occur is not standardized. So there's lots of standardization, lots of opportunity that we can work towards to better understand how to use whole genome sequencing data for surveillance, for outbreak investigations, and other public health and infection control programs. And then one piece that I think is very important is the experience, the infrastructure, and the data sharing. So I know I'm not going to spend enough time on this, but it's probably one of the more important aspects. So there's a wet lab, the hands-on technical aspect, and then the dry lab, so the informatics and pipelines and analysis. The experience that clinical labs have in each of those 2 areas, unfortunately, is still limited. It's being developed, I feel, and we're becoming much more adept at implementing these sorts of programs, but it still is something that is up and coming. So to improve and advance that infrastructure is important. And then the last piece is the databases and the data sharing. So we need to, as I started this talk, we need to be able to have a system that shares the genomic data between areas, between hospitals, between regions, between provinces, so that we can look for similarities and really use that data to its maximum. I feel that it's important that this data, for many reasons, is also available at a patient level in many cases, depending on what the importance, depending on the type of data, and that trying to test as close to the patient gives us potentially a better turnaround time and other advantages, but that means that we need robust data sharing methods and tools so that that information at one location can be shared more globally. And so with that, I hope that was a quick tour of some of the considerations for whole genome sequencing as it relates to public health surveillance and infection prevention and control, some of the methods, and hopefully I was able to explain some of the utility in different areas of this type of testing. It is a very powerful and important tool and will increase in use going forward. We always need to consider resolution and relatedness. So how do we interpret results, and how detailed do those results need? The other major consideration that I hope I was able to convey is that we need to have standardized methods to convey this information to the end user. So it's good if the lab can process data and report and develop these whole genome sequencing information, but without an understanding of what it means and without an ability to convey that complex information, it can go it doesn't have the same impact. And so finally, hopefully the last piece that I do want to end with, this information is only part of a puzzle. This is only part of the surveillance networks. It is still very much important, and the number one key for a surveillance network is the clinical epidemiological investigation. So clinical epi is needed to put all of this laboratory data into context. And with that, I'd just like to, again, thank everyone for joining today and pass it over to Vanessa for questions.
Thank you so much, Aaron. That was a fantastic presentation. So now we'll move on to the Q&A segment of this event to address some of the questions from our audience. And just a quick note to everyone, you can continue to enter your questions into the Q&A pod if you have not already had an opportunity to do so. And so we have a couple of questions already to get started, Aaron. And I think the first is has to do with WGS during the COVID pandemic. And I think you're very well positioned to answer this, as SickKids was part of the Ontario COVID Genomics Network. So the question is, what are some key learnings from using WGS during the COVID pandemic?
Thank you. So yes, we definitely used and continue to use whole genome sequencing data for SARS-CoV-2 sequencing. I think a couple key learnings that I would take away. One is timeliness. So timeliness of access to the data and timeliness of sequencing ability and capacity. So the further we moved away from a time of an incident or say a potential transmission and outbreak, while that data is still helpful in retrospect, it had less of an impact on the immediacy and changes in the immediate nature. So I think the biggest learning is trying to develop methods that are rapid, that are universal, and that are shared across different sites. And I think that would be the second piece. The second key learning is sharing of information between labs, between sites, so that we can have a network that is able to respond in a way that that is equal among all the different labs and all different sites.
And when you allude to timeliness, was the timeliness related to some of the challenges that you had highlighted in your presentation with respect to information sharing and just the technical and bioinformatic requirements for the WGS during COVID?
I think timeliness has a few points to it. One, for COVID, at least at SickKids, we became very adept in the technical aspect. We were able to have a genome within 32 hours, 24 to 32 hours. But the logistics of ensuring samples were together, ensuring data was together, ensuring all that information that doesn't fall necessarily on the technical hands-on within a lab is still so key in timely approaches.
So we have actually another question, and it's related to a question that I had actually, and it was if you can provide examples of how SickKids uses WGS for decision or support decision making. So the specific question was, how can we use your GWAS for diagnosis? Is SickKids using it for diagnosis? And just to add on to that, like, not only am I interested or would be interesting to hear about situations where it was helpful, wondering if you could also highlight situations where it wasn't helpful.
So I think there were two questions there. One, diagnosis, and I'm not sure if the question is more related to using, their next-gen sequencing can be used as a diagnostic tool as well. That is a separate consideration. Here at SickKids, we are beginning to use it as a diagnostic tool. But from a perspective of surveillance, I think yes, we did. What's nice is that we would be able to look at patients, staff, understand if there were multiple cases of SARS-CoV-2 in a certain area or within a hospital. We could, again, rule out transmission between cases. We could never definitively really show that transmission occurred when we had the same virus, but provide that extra information to say what we thought was a transmission event, we've shown that it is not. And so that's the real power that I think that comes from that resolution, that comes from whole genome sequencing data, where we can definitively say that cases are not related, and that can have a lot of importance at a hospital level. Again, it didn't necessarily affect us in this way, but it could, I could see, allow certain units to declare an outbreak over sooner, because now they have a case that they can say is not related. So it has lots of downstream benefits that can be realized.
Thank you. There's a question about more about relatedness, and I think this is a very important concept. And the question is, how are the cutoffs defined for when two subtypes are not related, and is uncertainty taken into account?
Great question. So essentially it's organism specific, and this is the biggest challenge that I see that we have in the lab going forward, to really define what is related. Every organism is going to be different. We can try to fall back to what's been provided within literature, which unfortunately is actually quite limited. And so these sorts of studies to determine what is related need to be continued. This is a very important, I believe, at least a very important area of continued research and continued activity to understand how organisms are related and how we can tell if two organisms are related from a genetic sequence. What we need to do, I think, is to look at outbreaks, to look at clusters, to look at patients over time and sequence multiple times the same pathogen within an organism or within a cluster and start to define it ourselves. So it's not 5 base pair differences. It could be 5, it could be 20, it could be 100. Every organism will be different, and it's something that I think still, like I said, is still something that needs a lot of ongoing work.
And maybe I can just add a little bit there, like not only like you need the technical and bioinformatic expertise, you need the expert like organism expertise to understand the organism characteristics and mutation rates in that organism. So it's definitely not a simple task. So we have a question about phylogenetic. This is a very good question. Can a phylogenetic tree incorporate epidemiological information in addition to genomic information, which would be very helpful?
Yeah, definitely. And I think the easiest way that this is often incorporated by adding information based on the name, sample names within a tree, or by colors or shapes. Lots of different ways that you can try to link clinical epidemiological information, units, households, whatever measure you are looking at, may be defined based on a certain color shape. So by using that information within a genetic phylogenetic tree, is one way I think that we could you could combine that data. And visually, it's helpful. I do find it's helpful as long as you understand how to interpret the genetic portion of the phylogenetic tree.
I agree. Okay, the next question is about a controversial topic. I'll leave it up to you whether or not you want to answer it. What is your opinion on whole genome sequencing in wastewater samples?
I'm not sure how to respond to that. So from a surveillance perspective, I think we're trying to, at least I was trying to really focus on how to define organisms within a within a cluster, within an outbreak. Whole genome sequencing within wastewater, I think serves a different purpose, part of surveillance possibly, but not necessarily something I could speak to.
I think that's a very good answer. Next question, again, just getting your thoughts on the utility of whole genome sequencing in early detection of novel emerging zoonotic diseases.
So I think whole genome sequencing is definitely an important tool. It depends on how we amplify, the wet lab portion. It will be important to understand a pathogen. It gives us a lot of information. Many times when we have say a novel zoonotic infection, we do have, or that maybe doesn't have a targeted assay, we may be able to fall back on more of a metagenomics approach. It's almost as a surveillance and in that way diagnostic tool, where we can use some alternate methods to try to find out what's there. Fortunately, I do think it's an important aspect, but not necessarily something that that we see every day.
So we have a question about, genotype and phenotype, and it's a good question. And the question is, would it not be better to combine WGS with other microbial characteristics, such as similar capacity to produce toxins, similar genotype and phenotypic characteristics?
So yes, I think it depends on what your goal is at the same time. So if we're wanting to understand how the genotype translates into phenotype, that's one potential question, and it might lead to a better understanding of the type of disease that we see. So we know that we may have genes that are not expressed in the same way. So we would want to understand if the change in that genotype is leading to a change in expression in phenotype. But from a surveillance perspective or an outbreak perspective, I think most of the time we're looking for transmission. So it depends on what the goal is. It's definitely useful in that regard, but it's ultimately what is the goal of the use of whole genome sequencing.
Yeah, I think that's a really, really great point, Aaron. And something, just to reiterate, like whole genome sequencing is a great new tool, but it's just one tool in our toolbox, and it's not the hammer that you can use to fix everything. Okay, a question about turnaround time. So turnaround time for WGS is, I think as you alluded to, has a significant impact on its use during outbreaks, particularly if there's a very long turnaround time. And I don't know if you can answer this question or not, but are there any plans in Ontario to invest in the technology so that it can be more timely, as you suggested, for determining the relatedness of sporadic cases? And the example here is for enteric organisms like Salmonella.
So I can't comment on any investment opportunities. I can just maybe reiterate that I agree that it's important to have a very as small as turnaround time as possible. And I think that's using the technology as close to the patient and as close to the sample as possible. There are lots of challenges to that. There's, again, the wet lab challenges and then the bioinformatics challenges. There are lots of different methods, I feel, where we can, again, work together to especially help with each of those. Sharing of information for the wet lab, sharing of procedures and protocols, and then from the informatics support, well, that may be decentralized. More sharing of protocols and pipelines and processes can also occur there. So I think there's lots of opportunity. I very strong agreement that it's important to have a rapid turnaround time to affect patient care as best as possible.
And I mean, like over the pandemic, there was a significant investment in Ontario for COVID genomics. So I think, I think the interest is there. I think we did well, and I think we can only do better moving forward. Okay, so there's a comment and a question again related to relatedness, and I think this is a really important topic that you brought up. So again, just reiterating the point that different organisms will have different rates of mutations, different molecular clocks. And so given the unknown, that this is unknown for many common antimicrobial resistant organisms, what do you feel is the best way to use EPI and WGS to determine relatedness? Like, how can an individual institution best contribute to defining what qualifies as relatedness? And are there data share mechanisms that would help to build these databases?
Great question. And I think I really like the idea of speaking to the data sharing and the databases. So unfortunately, now a lot of it is still through publication and through the literature. So looking at each individual organism one by one, presenting that data and genomic data over the course of multiple strains, multiple outbreaks, and within the same patient over time, and contributing to the literature is right now probably one of the best ways that this data can be disseminated. It really is still sparse, even for some of the important or common pathogens, MRSA, influenza, VREs. The data and how to define relatedness is still relatively sparse. So hopefully contributing to the body of literature and the evidence is really I think one of the best ways to continue publish, get the data out there so that we can use that knowledge within Ontario and not just Ontario, but within the scientific community.
So one last question about relatedness, and I think it underpins another great point about mutation rate. And so the question is about specific to organisms. So one, can you speak at all to Listeria, which can live in processing environments for very long periods of time? And conversely, about clustering perfringens that multiply so quickly, like how quickly would the genome be changing? And I like this question because it highlights the fact that you may know the mutation rate in an organism, but that may change depending on the environment.
I think that's a great question. I think that's essentially what it boils down to. It depends on how you're going to use the whole genome sequencing data, how you will look at the relatedness between organisms within each environment, and you need to define that up front. It's part of your clinical validation, it's part of your clinical determination of relatedness in order to understand each of those different environments and each organism. So I think that's just how we need to define that type of method.
Thank you so much, Aaron. And amazing questions from the audience today. We're out of time, unfortunately. So as we wrap up today's PHO Microbiology Rounds, I would like to sincerely thank Dr. Campagato for presenting, and I would also like to thank everyone who joined us for today's webinar. So you can expect to receive a brief and anonymous PHO micro-round survey for today's session. Please try to complete this as it really helps us to improve our programming. And as an important reminder to everyone to please save the date. Topic, the Ontario Public Health Convention will be held on March 26th and April 3rd in 2024, and is now now accepting abstract submissions for the program. So please check out tophc.ca for details. And lastly, to access past PHO presentations and to view confirmed and upcoming sessions, please visit the PHO website, head to education events, and click on presentations. So thank you very much again, Erin, today for joining us, and everyone for joining the session, and please have a wonderful day.